Yep, I've opened it. You can see it, right?
Yes, we can, but I can put it into the presenter mode.
Okay, there you go.
Yes, okay, yeah, it's good. So yeah, good morning to USA and good afternoon to the rest of the Europe. And my name is Dulmini Nanakar. I'm a research fellow at the Estonian University of Life Sciences, and it's my great pleasure to introduce today's guest lecture speaker, Professor Asgi Fazleabas from Michigan State University. So Professor Asgi is a globally recognized expert in reproductive biology, particularly in the area of uterine biology, implantation failure, and endometriosis. So, with the distinguished academic career, he has made a significant contribution to the understanding of maternal fetal interaction, and his research has been continuously funded by NIH since 1986. So, he has authored over 250 peer reviewed publications and developed a non human primate model that has greatly advanced research in reproductive health. So in today's guest lecture, he will present models for embryo implantation and decidualization, a role for notch. This fascinating topic explores the key molecular processes involved in primary pregnancy, focusing on how notch signaling influence both embryo implantation and decidualization. So we are excited to have him share his expertise, and I hope you will find this lecture both informative and inspiring. So please join with me to welcome Professor Aski. The floor is yours.
Thank you. Good morning, everybody. Nice to see everybody. So what I thought I'd do is just take you through how we've thought about notch as an important molecule with regards to its function. But just to give you an idea, this is where we work. This is the research building where we have our research, and that's our medical school. And this is called the medical mile. So it goes all the way up to all our hospitals and everything else. So for many years, I've really been fascinated with, let me minimize this so I can see the slide, if that's okay. Is that right? So for many years, I've been really fascinated with understanding how this little embryo arrives in the uterus and how does it send its specific signals at this very specific time during the window of implantation to allow for implantation to take place. And why is it that you only have this very limited window of time? And if implantation takes place outside of this window where it's receptive and the uterus can be selective as well as the embryo, you then have pregnancy loss. And so many years ago, we started thinking about how do we study this? Obviously, it's not something we can study in the human. So we developed a non-human primate model with the baboon. And the baboon is very, very similar to the human in terms of its cyclicity, its menstrual cycle, and also the hormones it produces in response to the establishment of pregnancy. Now, there's baboon chorionic gonadotrophin, obviously, but at the time of implantation, what happens is that the same embryonic signal that you see in humans is secreted by the developing embryo, and then it increases progesterone by rescuing the corpus luteum and maintains the production of chorionic gonadotrophin through the first part of pregnancy to maintain the corpus luteum before the placenta can take over. So we were really interested in this window of time, and we created a model to simulate pregnancy. So baboons also have a very low fecundity, just like women. And so it's very difficult to try to get implantation sites and even detect implantation sites early in the window. So what we did was to try to create the endocrine milieu that happens during this window of implantation. So in this, what we do is we put in a cannula that's embedded into the oviduct, and then exteriorize under the skin. So we can then come in with an all-Z mini pump and then release recombinant chorionic gonadotrophin in a pulsatile manner. And over a 24-hour period, this is the equivalent of what a baboon embryo would produce in vitro. So by monitoring the cycle, and then on the 6th day post-ovulation, we begin the secretion of chorionic gonadotrophin. And these 4 days of treatment mimics the window of time that the embryo is free-floating in the uterus. So it's about 30 IU per day, which is about the measurable amount of chorionic gonadotrophin that the baboon embryo produces. Like I said, it's the window of time when it's free-floating, and it's also the exquisite window of time that the corpus luteum becomes responsive. So following this treatment, what we did was first histologically analyze what happened, and what we were surprised to see was that each of the major cell types, the luminal epithelium, the underlying stroma, and the glandular epithelium, all responded very dramatically to this very short window of time of HCG treatment. What we see is that the luminal epithelium undergoes a plaque response, which is important for glycogen accumulation, as well as loosening the tight junctions so that the trophoblast can penetrate between the lateral membranes. The underlying stroma take on a myofibroblast phenotype, which is critical to initiate decidualization, and the glands become highly secretory. So in addition to this morphological analysis, what we also did was did an extensive array analysis and found that each of these different responses are associated with specific gene expression changes. And the one that we were most interested in was the change in Notch signaling at the maternal-fetal interface, as well as the induction of both alpha smooth muscle actin and Notch1 expression in the underlying stroma. And alpha SMA transcriptionally regulates Notch1. So these are two really early events that are associated with the decidualization process. And then what we saw was the glandular secretory activity was lots of histotroph secretion, along with emulatory molecules such as glycodelin and complement 3. So since we had seen these changes in both alpha SMA and Notch signaling, we started looking through the literature and saw that when we looked in mouse models particularly, that particularly at the time of adhesion and invasion, that Notch and its ligands were expressed both in the embryo as well as the endometrium. And if you then dissect these times out, you find that when the blastocyst is arriving for attachment and adhesion, you see that all 3 of the Notch ligands, together with 2 of the Notch 3 of the Notch receptors together with 2 of the Notch ligands are expressed in the embryo. The luminal epithelium only expresses the ligands so that there's interaction between the embryo and the endometrium. And then both the glandular epithelium as well as the stromal cells begin to express both the receptor and the ligand. So the first set of experiments we did to ask this question was under progesterone receptor Cree, which affects all progesterone responsive cells in the endometrium. We basically decided to knock out Notch1 in particular, because we had seen that Notch1 by immunohistochemistry and some of the other experiments showed the highest expression in both in the glandular epithelial cells as well as the stroma. And what we also did was look to see when Notch1 was expressed in all of our 3 models. So if you look at the mouse at embryonic day 6.5, you look at the human with HCG treatment, and you look at the baboon with HCG treatment, you can appreciate that the stromal cells, in addition to the epithelial cells, but the stromal cells in particular, begin to express Notch1. And then once we knocked out Notch1 under progesterone receptor Cree in the mouse model, what we found was that it mediates stromal cell survival, and that the absence of Notch1 results in decreased cellular proliferation, decrease in G1S checkpoints, and increase in apoptosis. So essentially, it inhibits the proliferation of the stromal cells and increases apoptosis. And for the initiation of decidualization, obviously, you want to inhibit apoptosis and increase cellular proliferation. But what surprised us was that when we looked at the Notch1 knockout, they had impaired decidualization, a decrease in the first litter size, but after that, these animals had no implantation or pregnancy failure. So the question that we wondered was whether there are potential compensation from the other notch receptors that allows for implantation to take place. Like I showed you in that initial summary slide, that notch 1, 2, and 3 are all expressed in both the embryo as well as the endometrium. So in order to address that question, we went back and looked at the notch signaling mechanism. So all of the notch receptors signal in this manner. So there are juxtacrine interactions. You have the ligand on one cell, and you have the receptor on the other cell. And there are two cleavage divisions that take place. What happens is when the ligand binds to the notch receptor, it activates two cleavage cascades. First, it releases the extracellular domain from the transmembrane domain through ADAM10 interactions. And then in the second part of the cascade reaction, gamma secretase then clips the transmembrane domain to then release the intracellular domain. And the intracellular domain is really the main component of notch signaling, which then moves into the nucleus, and then by binding to RBPJ or CSL, RBPJ in the mouse, CSL in the human, and recruiting other co-activators along with mastermind will then initiate transcription. So since all of the notch receptors work in this manner, we decided to look at CSL or RBPJ in the mouse. And previously, a group had shown that if they had done in situ hybridization for RBPJ, what you find is that RBPJ expression during implantation in the mouse absolutely parallels Notch1 expression that we had shown in our work. So then to ask the question whether notch signaling is really critical, what we did was under progesterone receptor Cree, we deleted RBPJ exon 6 and 7. So we created a mouse that now had no RBPJ signaling, which means they could not activate any kind of notch signaling. When we looked to see what happens with these mice, what was interesting was initially they are subfertile. So when we did a study for looking at overall pregnancy over a 6-month study, we found that there was a substantial decrease in the number of pups per litter, as well as the mean number of litters per mouse in the RBPJ knockout. But as we followed these animals in a 6-month breeding trial, the first litter was perfectly normal. We saw a decrease in the second litter, a more pronounced decrease in the third litter, and after that, in the rest of the 3 months, these animals become completely infertile. So it seems like there's this very nice balance between what's happening with notch signaling as well as implantation. So our question was, what is it that's happening progressively in these animals that then allows them to get infertile over time. And in collaboration with my colleague Ripla Arora, we started looking at three-dimensional mapping of the endometrium in the control as well as the RBPJ knockout. Now, she has shown that during the establishment of pregnancy, the endometrium in the mouse has these beautiful folds that are needed to be developed to allow for the implantation to take place in the implantation chamber. But when we looked at what happens with our RBPJ knockout, we do not see these transverse folds that we are expected to see. Instead, we see the longitudinal folds. So the endometrium itself is undergoing a process that's not conducive to implantation. And then when we look at the embryos within these longitudinal folds, instead of the embryo going into the implantation chamber, it was attaching parallel to the implantation chamber. So in the absence of these transverse folds, we are not able to create the implantation chamber. As a result, the embryo is disoriented and is then trying to attach in a longitudinal as opposed to a mesometrial, anti-mesometrial way that it normally has to happen for implantation to take place. So this is our first indication to suggest that in the absence of overall notch signaling, the endometrium itself cannot organize itself appropriately to allow for appropriate implantation to take place. Furthermore, what we were also able to see is that when we looked at these animals over time, at the end of pregnancy, and that's data I'm not going to show you, but I'll tell you about it, is that the nodules that are normally seen at the implantation sites and have to undergo degeneration for another implantation to take place never disappeared in our RBPJ knockout. And what we found was when we looked at the nodules postpartum, we see a huge influx of macrophages and immune cells into this area, plus the normal repair between the epithelium and the myometrium, which normally takes about 3 days postpartum in the mouse, is delayed for about 10 days. So there are a couple of things that are happening early in pregnancy where the embryo cannot orient properly, and that's over time leads to infertility. And over time, when the animals continue to try to get pregnant, the nodules persist. And as a result, because of inappropriate epithelial myometrial repair, they can no longer implant. Then our also question was, how important is the luminal epithelium? So in this case, what we did was we used the lactoferrin Cree, and under the lactoferrin Cree, we also knocked out RBPJ. And to our surprise, there was no impact of epithelial, where I'll show you something when we overexpress, it's very different. But if we just knock out RBPJ in the epithelial cells, both glandular and luminal epithelial cells, using the lactoferrin Cree, these animals are perfectly fertile. They do not have any issue with getting pregnant. So in this case, it appears as far as notch signaling is concerned, it's the stromal cells that are important, and perhaps really is the early decidualization response. When it's inhibited, these animals cannot get pregnant, and the stromal cells, which my colleague Ripla has shown, she actually has a paper out just now on the role of COX. It's in bioarchives just today, showing that there is this very interesting interaction using three-dimensional analysis between the epithelium and the stroma. So perhaps it's a stroma that's now regulating the orientation of these embryos, and as a result, these animals cannot get pregnant. Now, in our other studies, what we had also shown was that notch needs to be very tightly controlled for decidualization to take place. So notch is important for proliferation, initiation of differentiation, but if notch persists, the cells will not differentiate. So we had shown in some of our original in vitro studies that if we overexpress Notch, the cells in vitro would not decidualize. So Notch has to undergo this initiation early in pregnancy and then has to disappear to allow decidualization to be completed and for pregnancy to be established. So based on this, we then decided to ask what happens in our animal models if we now overexpress the intracellular domain of Notch. So this is the major component of Notch that's involved in cycling. So what we did in this group of animals here, again under progesterone receptor Cree, we overexpressed the intracellular domain of Notch. So these animals are now overexpressing Notch. And what we see is, you can see by GFP that the Notch is overexpressed, and this can also be measured by PCR for GFP protein and the intracellular domain of Notch. And when we do this, what we find is these animals are completely infertile. It doesn't matter whether it's heterozygous or homozygous mouse. These animals cannot get pregnant at all when we overexpress Notch. In addition, what we find is the overexpression of Notch also inhibits the size of the uterus. So the uterus in these animals are much smaller, suggesting again that they are not undergoing that differentiation during the postnatal period that allows this uterus to grow and allow it to become mature enough to maintain a pregnancy. And when we looked at the progesterone target genes, what we find is in our animals at 3.5, when we overexpress Notch, the normal increase in all of the progesterone target genes, Indian hedgehog, HOXA10, amphiregulin, all of which go up during early pregnancy, are now markedly diminished. Instead, what we find is that the estrogen target genes, which should normally go down, are overly expressed inappropriately at this point of pregnancy. So you see an increase in C3, MUC1 and MUC4 are highly upregulated, as well as LIF, which continues to be upregulated. Even though LIF is really important for early implantation, it continues to be overexpressed. So we're seeing a dichotomy again, and progesterone is a regulatory component for Notch regulation, and the overexpression of Notch is not allowing for the progesterone target genes to be expressed. And the other thing that we see associated with the small size of the uterus is that these animals have no glands. And so this again relates to the importance of Notch and its regulation for differentiation. And progesterone receptor Cree usually turns on at day 3 postnatally. And this is about the time when you begin to see the adenogenesis and the differentiation process. And the overexpression of Notch does not allow these glands to develop. Instead, what we see is that the normal glandular markers, like FOXA2, continue to be expressed in the luminal epithelium, suggesting that in the luminal epithelium cannot invaginate, form the neck glands, and essentially develop the glands. And we see that both by in situ for LIF, the epithelial cells only express LIF in the overexpression mouse, as well as the luminal epithelium continues to express FOXA2 inappropriately in these animals. And then if you study the implantation side of these animals, here again, you'll find that they basically do not have the normal blue dye. The normal implantation is not seen. And instead, what we see is that the embryos are free-floating within the uterine cavity. And perhaps because of the overexpression of MUC1 and MUC4, cannot attach and invade into the endometrium. And in the absence of glands, obviously, again, this is very similar to work that had been done in the sheep, where giving progesterone early postnatally blocks glandular development, and the embryos, the conceptor cells, are seen to be free floating. So essentially, in the absence of glands which do not differentiate because of the overexpression of Notch, the embryos cannot attach for a multitude of reasons. And we went through a complete study of trying to understand what happens. And what we were able to find is that we see an increase in PU1, which is a transcription factor that upregulates DNMT3. So there is DNA methylation, and as a result, there is epigenetic suppression of the progesterone receptor in response to the overexpression of Notch. And so overall, when we overexpress Notch under progesterone receptor Cree, we have glandular failure, which also results in implantation failure because these animals cannot implant in the absence of glands. What we find is that inappropriate upregulation of the estrogen receptor, estrogen-regulated genes, and a downregulation of progesterone-regulated genes, as well as the progesterone receptor, which is silenced epigenetically. And as a result, decidualization also fails because we find that progesterone receptor, Indian hedgehog, COOP-TF, and BMP2, all of which are important for decidualization, are all downregulated in response to the overexpression of Notch. So essentially, it's a very tight balance associated with Notch, where it has to come on early. If it doesn't come on early, you have implantation failure. And if it persists, you then have complete pregnancy failure that occurs in these animals right from the very onset. So since we had also seen that luminal inhibition of RBPJ, which blocks all of Notch signaling in the epithelium, we also then asked the question, what happens if we now overexpress N1-ICD only in the epithelium? So then we went back, and again, using the lactoferrin Cree, we overexpressed Notch1. And here again, what we find is, unlike knocking out RBPJ, in this case, if we overexpress Notch1 in the epithelium, these animals are also infertile. And looking at what happens with these animals, you find that there's absolutely no implantation in these animals. It cannot be rescued by the injection of LIF. But interestingly, if we now take away the ability of Notch to signal, that is by mating these animals to the RBPJ knockout, which is taking away Notch, the inability of Notch to work through RBPJ, we find that there is a rescue, and these animals can now implant. So it's perhaps two different processes that are occurring between the stromal cells and the epithelial cells, and something that we're trying to address, whether this could potentially be non-canonical Notch signaling that might be then enhancing the ability of Notch to compensate, this overexpression of Notch to compensate by taking away its ability to signal using the RBPJ, the canonical pathway. So these are studies that we're trying to do to try to understand what might be happening between the canonical and non-canonical way of Notch signaling. But we also see an aberrant changes in these animals. So if we now see that if we overexpress Notch under lactoferrin Cree, you can see again by 3D analysis that the normal glandular formation that has to occur does not occur in these animals. So again, epithelial overexpression affects the ability of the luminal epithelium to invaginate, form the endometrial glands that are important for implantation. And again, if you look at the luminal epithelium, it's completely disoriented. You do not see these really nice transverse folds, and instead you see this sort of mix of transverse folds and potentially invagination, but not appropriate morphological components that are critical for the embryo to implant into the implantation chambers. So the whole luminal epithelium and the glandular epithelium is completely messed up when we overexpress Notch1 specifically in the epithelial cells. But interestingly, of course, when we look at both progesterone and estrogen-regulated proteins, we see a similar response in these animals, in that the estrogen-regulated proteins, again, are upregulated, and the progesterone-regulated proteins are downregulated. So what we've been able to show, and then with RNA-seq, we find that there are differences between the luminal epithelium and the glandular epithelium, in that when we isolate each of these different cell compartments, there is an increase in cell cycle genes and cell proliferation in the presence of overexpression of Notch. And whereas the glands hyperproliferate and increase their estrogen sensitivity at the inappropriate time. So to summarize what I've been trying to show you with all of these model systems is that in the presence of luminal overexpression, what we find is that this normal invagination that's required for lymph secretion to make sure that the embryo can implant is disrupted. The luminal epithelium does not differentiate appropriately. The glandular epithelium hyperproliferates, responds to estrogen, but also does not release lymph, and lymph treatment does not rescue this process. And instead, what we find is we have implantation failure because overexpression in the epithelial cells does not allow for this normal differentiation to take place that's important for implantation. So those are some of the studies that we've been doing using different animal model systems, particularly the mouse and the transgenic mouse systems, to try to understand the role of Notch in both early pregnancy, implantation, as well as Decidualization. So the next question we had was, since we've shown that Notch is so important for Decidualization, what then happens to these stromal cells with regards to the Decidualization process? So we went back and looked at each of our models, and before I said, this is, you guys all probably know this, but that Decidualization is really important for promoting embryo implantation, controlling trophoblast invasion, and allowing for the fetal allograft to be accepted, particularly in mice. And in rodents, Decidualization is physically induced by the implanting embryo. Human Decidualization is much more complex. We just think that it's upregulated by the increase in progesterone. But I think there's a lot more complicated effects associated with the process of Decidualization in the human and the non-human primate. And we already know that impaired Decidualization can result in long-term effects later on in gestation with regards to preterm labor, preeclampsia, and IUGR. So in order to really begin to think about Decidualization, we went back to each of our models. This is the RBPJ knockout, the overexpression of N1-ICD, both in the glands and stroma, as well as in the epithelial cells. And using the artificial Decidualization model, where you prime these animals with estrogen and progesterone, and then you either inject oil or scratch the uterus and look to see what happens 3 and 5 days later, you can see that in each of our models where you block Notch signaling, and I already showed you that even the Notch1 knockout has Decidualization failure, but if you block all Notch signaling, or you overexpress N1-ICD in the stroma or the epithelium, you find that these animals cannot respond to artificial Decidualization. We see a decrease in the decidualization markers, Wnt4 and BMP2, and a significant downregulation of progesterone-regulated genes in these animals. So our interest, as you also know, is in understanding what the impact of decidualization is with regards to early implantation and the human situation. And I've told you that notch is very tightly balanced during this process during implantation. So using human stromal cells, we ask the question again, how critical is the early expression of notch during the decidualization process? So in this case, using siRNA, what we did was we inhibited notch expression in endometrial stromal cells prior to the initiation of treatment. So we use estrogen, progesterone, and dibutyrocyclic ANP. So we wait 3 days after we treat these cells with the shRNA, and then treat them with estrogen, progesterone, and cyclic ANP, and then look to see what happens 6 to 8 days later. And what you find is if you pre-treat these cells by knocking out notch, that the normal decidualization response, which is characterized by the expression of both IGF-BP1 and prolactin, is significantly downregulated in these cells. However, as I told you, that notch has to be decreased to allow for decidualization to continue. In this case, if we now first treated these cells after 5 days of culture with EPC, and then transfected in the shRNA for notch, what we find is that we downregulate notch, but there's no effect on decidualization. So these cells, once they begun their initiation and their transformation, it doesn't matter whether we take away notch, they still undergo their decidualization response, suggesting again this balance of notch for early responses versus the absence of notch in late responses. So I've shown you this slide before. We see it in all of our 3 species that we work with. And what we think is that notch signaling is very, very important early for transforming this undifferentiated phenotype, the stromal cell, into the pro-inflammatory phenotype that is characterized by the expression of notch alpha SMA and a myofibroblast phenotype, and that notch signaling has to be downregulated to allow for this cell to now go from its pro-inflammatory phenotype to its secretory phenotype, which is the fully functional decidual cell. So this kind of summarizes things that we've done over the years. In the human model, chorionic gonadotrophin can act on these stromal fibroblasts to increase Notch1. Progesterone interacts with Notch1 to upregulate these genes that are important to inhibit apoptosis and initiate differentiation. So my lab has also been interested in decidualization and its consequences with regards to pathologies in the human. So we know from a number of studies that people have published, and this was a review that Carlo Ticconi and I did, that we find that disordered decidualization response can result in a large number of changes, including senescence, immune dysregulation, a large number of changes that are important for the establishment of pregnancy. And that there are diseases like endometriosis, which is what my lab focuses on, and consequences of disordered decidualization that can result in late pregnancy loss because of various components such as fetal growth retardation, preeclampsia, and placental response, and all of them being critical for affecting either recurrent implantation failure or recurrent pregnancy loss. So our first question was, well, what happens with endometriosis? We know endometriosis does contribute to infertility. And when we went back and looked again in our baboon model, and over the years we've developed a baboon model to mimic endometriosis, where we can inoculate these animals with menstrual tissue and then follow them. And these animals, like women, also get spontaneous endometriosis. And if you now look at the baboon endometrium in animals with spontaneous disease versus controls, what we find is that two of the Notch receptors, particularly Notch1 and Notch4, together with its ligands and its downstream genes, are all downregulated in the presence of endometriosis. And this is also evident in women with endometriosis. And you can see that if you look at the endometrium of women with endometriosis, again, we see a very, very similar response to what we see in our baboon model. This is by PCR. And then you can see that if you do the immunohistochemistry, you look at the eutopic endometrium of women with endometriosis, you see that both Notch1, Notch4, and the downstream target HES5 are all downregulated in the endometrium of these women that have endometriosis, which can then contribute perhaps to Decidualization failure. So what then could be doing, what is the upstream regulator that might be affecting Notch1? So SIRT1 can negatively regulate Notch1, particularly the N1-ICD expression, and it does that in response to the ligand stimulation. I showed you that these are all downregulated. And when we then looked at both our baboon model and our humans, what we find is that SIRT is highly upregulated in the endometrium of women and progressively in baboons with endometriosis. The advantage we have with our baboon model is we can harvest endometrium during the course of the disease. And you can see here that as the disease progresses, where we see that Notch1 goes down, you see an increase in SIRT, which is comparable to what we see in women with endometriosis. So this is just summarizing a lot of work that we have done. What we think is happening normally is that in response to Notch, in the normal physiological situation, it increases FOXO1, IL-8, and a number of other early response genes that allows for the initiation of Decidualization. And this then allows these decidual cells to express both prolactin and IL-15, which is important to enhance the ability of the NK cells, particularly uterine NK cells, to clear the senescent cells, because during this process, you get both senescent decidual cells versus the normal decidual cells. Now, in the context of endometriosis, what happens is we see the decrease in Notch signaling, but we still have an open question as to how this then directly regulates FOXO1. But suffice to say that if we look downstream, we know that FOXO1 and IL-8 is downregulated. So as a result, because the early response genes are downregulated in response to the decrease in Notch, the normal Decidualization response is inhibited. As a result, IL-15 is not secreted, and now you get an increase in senescent cells that are inappropriate and cannot sustain a pregnancy. So in order to ask whether that is real, we went back again and looked at the endometrium of women with endometriosis. And you can see that if you look at lamin B1, which is a marker of senescence, it's markedly downregulated in the eutopic endometrium of women with endometriosis, suggesting that this is one pathway that might be affecting Decidualization. And then if we go back into our stromal cells again and knock down Notch1, we see that we can now induce senescence in these cells. And if you look at lamin B1 and P21, which are both markers of senescence. P21 goes up in senescent cells, lamin B1 goes down. And in the absence or the suppression of Notch in vitro, you see that we see these cells undergoing senescence, which suggests that perhaps this decrease in Notch1 might be contributing to inappropriate decidualization and enhanced senescence. So this is what we are working with right now. The increase in SIRT1, we still need to find out how is SIRT1 upregulated, but we know it can transcriptionally repress Notch1. And in the context of endometriosis, where we see the increase in SIRT1, we see a decrease in Notch1, and as a result, inappropriate decidualization. But we also see an increase in cellular senescence. So right now, a lot of our work is focused on trying to understand the mechanisms associated with cellular senescence in terms of both disease and normal physiology. So the other effect of disordered decidualization is recurrent implantation failure or recurrent pregnancy loss. So in order to do that, we went back again and got tissues from women that have recurrent pregnancy loss. And this is in collaboration with Carlo Ticconi at the University of Rome, who does a lot of recurrent pregnancy loss work. And we first looked to see what happens in the unexplained recurrent pregnancy loss patients. And what we find is that RPL, which we know is important for the initiation of decidualization, is markedly downregulated. And instead, interferon gamma that enhances inflammation is markedly upregulated in the endometrium of these women that have had 2 or more consecutive clinical losses and are thought to be associated with recurrent pregnancy loss. But what is then upstream of this? So EGFL7 is an activator of Notch. And now if we look at the expression of EGFL7 in either the secretory phase or the proliferative phase in these women that have recurrent implantation failure or unexplained recurrent pregnancy loss, we find that there is a significant downregulation of EGFL7 in these women, again suggesting that upstream activators of Notch might be disrupted, allowing for impaired decidualization. And again, you can take stromal cells from these women that have this failure, induce them to undergo decidualization. And interestingly, what we find is that it's only in the women that have unexplained pregnancy loss that we see a substantial inhibition of decidualization. Instead, in the recurrent implantation failure women, we see actually an increase to some extent of the decidualization response, which is interesting because Jan Brosen's group has shown that these women with recurrent implantation failure are superfecund and that they actually have a much more robust decidualization response that allows them to implant all embryos. It doesn't do the selection. And as a result, all embryos implant, but they undergo implantation failure because of premature upregulation of decidualization. And perhaps this EGFL7 in some way is altering the decidual response in women for implantation failure and suppressing the ability of the cells to transform in women with unexplained pregnancy loss. So to summarize that, what we see is that the normal response with PGE2 binding to its receptor increases dibutyrocyclic ANP, which can upregulate EGFL7, and EGFL7 can then activate Notch to allow it to undergo these cleavage divisions because it's soluble, and then allow for the intracellular domain to migrate into the nucleus and initiate Notch transcription. But in the context of unexplained infertility, unexplained pregnancy loss, this process in some way is inhibited. We don't know again whether this pathway is altered, but we know that somehow or other EGFL7 is downregulated. And as a result, this downregulation doesn't allow for appropriate notch signaling, and this inhibits the decidualization response that then could contribute to unexplained pregnancy loss. So what I hope I've showed you is that by using these different model systems, we've been able to try to dissect out what's happening with the notch pathway. And now most of our questions are really trying to understand the upstream, downstream mechanisms that are associated with this process. So my final slide is really the people that have done all the work. The original notch work was started by an MD PhD student in my lab, Yalda Afshar, who's now an associate professor at UCLA. All of the RBPJ work was done by another MD PhD student, Mike Strug, who's just finished his fellowship in reproductive endocrinology at Stanford. And all of the overexpression work was done by Renvi Su when he was a postdoc in my lab, who now has his own lab in China. And all of the other work, Jenna Moldovan, who just graduated, is now doing a postdoc, did all the work on the RBPJ in the epithelial cells. Yong Sun has done all of the senescence work. And I didn't get a chance to talk about the work that's being done by Ariadna, Greg, and Niraj, which are really more focused on transcriptomics and microRNAs. And then a lot of our animal work couldn't be done without Sam and Erin, who run all of this. So with that, hopefully I've given you a little bit of an idea of what we're doing in my lab, and I'll be happy to answer any questions.
Yes, Professor Aski, thank you very much for the very interesting talk. Yes, we have quite a few questions. Subhashini, please go ahead.
Thank you very much, Professor Aski. It's a beautiful story. So I understand that notch signaling is very critical in initiation of the pregnancy and its persistence over time. Am I correct to understand that? So you said notch signaling is initiated mainly by HCG because you mimic the presence of the embryo using HCG only, and then came to this idea that notch signaling is mainly initially mediated by HCG alone, or other.
So yeah, I mean, HCG, we were trying to mimic what happens during early pregnancy.
Mm-hmm.
And so it's, there are probably other factors that come from the embryo, but in our hands, we really tested HCG first. And what we find is that chorionic gonadotrophin infusion initiates the expression of Notch1 in the stromal cells. So it's always in the glandular epithelial cells during the cycle. It goes up during the secretory phase, but for it to initiate its expression in the stromal cells, that happens in the presence of an embryonic signal. And you also see that in the mouse, that if you look at the implantation site, you begin to see the expression of Notch1 and RBPJ first right at the point of attachment, and then it spreads to the primary decidual zone. And then as the primary decidual zone becomes differentiated, and that in situ that I showed you, then it moves out to the secondary decidual zone. So it's those early stromal cells that are undergoing that initiation of differentiation that have to express Notch. And then Notch has to come down for these cells to completely transform into a decidual phenotype.
So have you tried like adding whole secretome of the embryo and seeing whether notch signaling get activated?
No, we haven't done any of those because, obviously getting embryos is tough.
Yeah, because I was thinking that, I don't know, you were ever interested about exosomes. So these embryos also secrete exosomes.
Right.
So and then we looked at this endometrial epithelial cell changes, and I never detected this notch signaling, but it seems so important. So I was wondering.
Yeah,
Is something
if it's happening, it'll be in the underlying stromal cells.
stromal cells only. Yeah,
Yeah.
okay, yeah.
Yeah. And we haven't done any exosomal
Do you
work.
think that so anyway, this embryo signals is first received by the upper layer, luminal epithelium, right? Do you
Right.
think that changes in that later courses, signaling, not signaling initiation in the stromal compartment? It could be also a possibility, right? Even you
It
don't
is.
see. Yeah, yeah.
It is. It is
Yeah,
definitely
yeah.
a possibility because, like the
Yeah,
second or third slide that I showed you with the morphological changes,
yeah, yeah,
when the luminal epithelium undergoes a plaque response, the underlying stromal cells initiate their myofibroblast differentiation. Now, if you look at our animals that we induce endometriosis and treat them with chorionic gonadotrophin, that response is muted. And so as a result, they don't differentiate. And in response to your question, we haven't done any of the secretome, but in Yalda's original paper, we did treat these stromal cells with chorionic gonadotrophin in the presence and absence of progesterone receptor antagonists. And what we find is that there is a synergism between progesterone
Okay.
and chorionic gonadotrophin to allow these cells to undergo decidualization.
So there could be other factors that actually contributes to.
Sure.
Okay. Thank you very much. Nice. I can read more.
Suranga, please wait before we go to your question. We have one question in the chat. So it says, like, is there a specific time frame when the rescue must happen to reverse the effect of notch impaired signaling, or it has to happen before fertilization?
We've never looked before fertilization. For our focus, we've always been early implantation. So I'm not sure I understand the question about fertilization. So if you looked at these animals, or even humans during the cycle, most of the notch expression is in the epithelial cells. You only see the expression in the stromal cells at the initiation of implantation. So it's much further down.
Okay, I hope Marilyn got the answer. If not, you can speak out if you still.
If they're interested in ovulation and ovarian function, there's lots of lovely papers with the role of Notch2, particularly from Kelly Mayo's group, showing that Notch signaling is critical for oocyte development and follicular genesis and stuff like that. But we've never studied that. That's all been done by other people. And Kelly Mayo's group was the first one that did a lot of that, and particularly focused on Notch2.
Okay, thank you. And Suranga, please go ahead.
Thank you, Professor Aski. It was very interesting. I'm just curious because if you look at the human cycle, even without an embryo, it's evident that the endometrium can undergo decidualization, right? As a part of, even people claim that is one of the characteristic feature of the window of implantation. Even I know a particular group is arguing in that way. So what could be the different, like I really didn't get that with the presence of embryo and the absence of embryo. If you look at these two scenarios, would that be the same?
Yeah. So in the human, of course, that around the spiral arteries, late in the secretory phase, outside of the window of implantation, is just before progesterone comes down. You see the cells around the spiral arteries undergoing that what we call a predestival response.
Yeah.
And that's unique to the human.
Yes, exactly. That's what I was asking.
Yeah. In a non-human primate, you don't see that. You only see the upregulation and the initiation of differentiation in the presence of an embryonic signal. And it's the same thing in the rodent. So is it, I always think about, and all of the other work we had done with regards to changes in the endometrium, comparing the human and the non-human primate. My take on that is that the human is so invasive that, to give you an analogy, it almost seems like the car has to be in fourth gear And when the embryo is there, it needs to kick into fifth gear to allow implantation to take place.
Because
Whereas in
of
the other species, because it does not have as invasive an implantation, the car can stay at neutral and then kick in the gears when the embryo is present and implantation starts to take place. So it might be an evolutionary adaptation in the human because of its invasive nature that the endometrium almost always has to be prepared. And that's probably why, implantation failure, recurrent pregnancy loss, and things like that are so much more prevalent in the human population than it is in the other primate populations.
Okay, so is there any more questions to Professor Aski? Seems no. Then thank you very much, Professor Aski, for your insightful lecture on this not signaling in embryo implantation and decidualization. And so we greatly appreciate your time and expertise today for this guest lecture. And so on behalf of Fazleabas and everyone that attend today's guest lecture, we wish you continuous success, and thank you very much for your time and all the patience and everything.
Okay.
And thank you. Yeah.
My
See
pleasure.
you
It was
all.
nice seeing you
Yeah.
all. Take care.
Take care. Bye.
Bye.